Hello everyone, welcome to QAxon. So in this video, you will learn about neuromuscular transmission. Under this topic, I will first start off with the structure of the neuromuscular junction, and then we will look at the steps in neuromuscular transmission. So here we go. Let's begin by understanding the structure of the neuromuscular junction. Now remember that this is a microscopic structure. It is not visible macroscopically. What is the neuromuscular junction? This is where the signal from the motor nerve fiber is transmitted to the muscle fiber. And remember that it is a 1:1 transmission, meaning that one motor nerve fiber will transmit its signal only to one muscle fiber. What is the challenge at this junction? The challenge is that there is actually a physical gap between these two structures, between the nerve fiber and the muscle fiber. So electrical signals cannot be transmitted across this gap. How is this challenge overcome? So the electrical signals are briefly converted into chemical signals and then again into electrical signals. And what is the chemical? The chemical is a neurotransmitter which acts by binding to its specific receptors on the muscle fiber. Now let's try and understand the structure of the neuromuscular junction using a diagram. In this diagram, you can see that broadly the neuromuscular junction is divided into three portions. The first portion is the axon terminal. As you can see, it is expanded at its tip to form the synaptic knob. Now this is the physical gap that I was talking about, and this is called synaptic cleft. Finally, you have the portion of the muscle fiber, the sarcolemma, which is called the motor end plate. You can see that it is thrown into many folds in order to increase the surface area. Now let's examine the axon terminal in more detail. You can see that it is full of neurotransmitter-filled vesicles. Now, in case of the skeletal muscle, this neurotransmitter is always acetylcholine. You can also see that there are many voltage-gated calcium channels on the membrane. In addition, there are also many mitochondria because this is an energy-requiring process. Now, when a signal is transmitted, the neurotransmitter, that is acetylcholine, is released into the cleft and then it binds to its specific receptors present on the motor end plate. These receptors are called nicotinic acetylcholine receptors. Now, in truth, they are nothing but sodium channels which open the moment acetylcholine binds to the receptor. So they are examples of ligand-gated sodium channels. In addition, you can also see that there is an enzyme in the synaptic cleft called acetylcholine esterase. Now the function of this enzyme is to degrade any acetylcholine which is present in the synaptic cleft. So this question is to test your short-term memory. Name the neurotransmitter released at the neuromuscular junction of a skeletal muscle fiber and name its receptor. I hope you remember that. It is always acetylcholine which is released at the neuromuscular junction of a skeletal muscle fiber, and the receptor here is the nicotinic acetylcholine receptor. Now, before we venture into the process of neuromuscular transmission, let's also understand what the motor unit means. A motor unit is defined as one alpha motor neuron and all the muscle fibers supplied by it. Now, this diagram will help you understand the concept better. So let's imagine that this is a muscle. For example, the biceps. And again in this diagram, it is the same biceps. Now you can see that in the first diagram, we have only represented a single motor unit. So you have an alpha motor neuron in the spinal cord, and it is supplying this group of fibers, muscle fibers, within the biceps. So this represents one motor unit. But here you can see we have represented two motor units. So this is one alpha motor neuron. It supplies one group of fibers within the biceps. This is another motor neuron, and it supplies another group of fibers within the biceps. So there are two motor units here. That means each muscle is made up of several motor units. And it also means that when you contract a muscle, it is not necessary that the entire muscle is contracting. A group of motor units may be contracting. So if you have to bring in more power, you're lifting a very heavy object, you might use more motor units. If you're just lifting a pencil, you might use a lesser number of motor units. So now let us come back to the process of neuromuscular transmission. We've already examined the three components of the neuromuscular junction, namely the prejunctional membrane or the axon terminal, the synaptic cleft, and the postjunctional membrane or the motor end plate. Now let's use the same diagram to understand the sequence of events in neuromuscular transmission. So we already know this is the axon terminal, this is the synaptic cleft, and this is the motor end plate. So during neuromuscular transmission, a signal, that is the action potential, arrives at the axon terminal, and that leads to depolarization of the membrane here. So because of depolarization, there is opening of the voltage-gated calcium channels. So calcium ions enter into the axon terminal. What is the role of calcium? Calcium causes an important process called docking. Just like how ships are docked, the same way calcium causes movement of these neurotransmitter vesicles towards the cell membrane. So there is docking of the vesicles towards the cell membrane. Also, these vesicles now fuse with the cell membrane, and by the process of exocytosis, they release the acetylcholine into the synaptic cleft. So calcium is important in docking and exocytosis. Now what happens to the released acetylcholine? Most of it binds to receptors on the motor end plate. We already saw that these are called nicotinic acetylcholine receptors. So when acetylcholine binds to these receptors, there is a conformational change in these receptors. Therefore, there is opening of sodium channels because these are ligand-gated sodium channels. So once these channels open, sodium ions abruptly enter in large amounts into the muscle fiber. So that leads to generation of a small potential here because there is depolarization. That small potential is called end plate potential. Usually the end plate potential reaches threshold and that gives rise to an action potential. So we now have a muscle action potential. So what was initially a nerve action potential has been translated into a muscle action potential. And what happens to this? The muscle action potential moves along the sarcolemma and is transmitted along the sarcolemma. So to repeat the steps involved in neuromuscular transmission, the first step is the arrival of the action potential at the axon terminal leading to depolarization. So this causes opening of voltage-gated calcium channels here, and there is influx of calcium ions. What do these calcium ions do? They cause docking and fusion of the neurotransmitter vesicles with the cell membrane. Then there is exocytosis leading to release of the acetylcholine into the synaptic cleft. And this acetylcholine binds to nicotinic receptors present on the motor end plate. Now, the binding causes opening of ligand-gated sodium channels leading to entry of sodium ions, generating what we call as an end plate potential in the muscle fiber. So this end plate potential often reaches threshold and gives rise to a muscle action potential, which is in turn conducted along the sarcolemma of the muscle. Now, a note on acetylcholine vesicles. Under normal circumstances, we never run out of acetylcholine, and this is because we are provided a surplus of acetylcholine at the neuromuscular junction. Now, to look at the figures, we have about 3 lakh vesicles per axon terminal. And within each vesicle, we have about 10,000 acetylcholine molecules. So that's a lot. And during each nerve action potential, only 60 to 120 vesicles are released. So this means a normal person would not run out of acetylcholine. Now, acetylcholine is released in a quantal fashion, which means at a given time, a quantum of acetylcholine is released. Acetylcholine is synthesized in the soma and then transported to the axon terminal. So the enzyme CHAT, that is choline acetyl transferase, is involved in the synthesis. So acetyl-CoA combines with choline in the presence of this enzyme to generate acetylcholine. Now, once acetylcholine is released, the free acetylcholine in the synaptic cleft is degraded by the enzyme choline esterase. Now, in case any acetylcholine leaks from this cleft into the blood circulation, it is degraded by the enzyme pseudocholine esterase, which is present in blood. Now, though we discussed the process of neuromuscular transmission, how is this process brought to an end? How is it terminated? That is important because otherwise we would have sustained stimulation of the muscle, which we do not want. Now, what normally happens is the free acetylcholine present in the synaptic cleft is slowly degraded by the enzyme choline esterase. So the concentration of free acetylcholine in the cleft falls, and this leads to dissociation of the acetylcholine that is bound to the receptors on the motor end plate. Now, the moment the acetylcholine dissociates from its receptor, the sodium channels close, and that leads to termination of the muscle action potential. So this means that the enzyme choline esterase plays a very important role in the termination of the muscle action potential. Now, I will conclude this session with a small question for you to explore. And the question is, what is miniature end plate potential and why is it significant? With that, we come to the end of this session. Thank you.